Hi, my name is Zheng Wei. I am a postdoc of Department of Automation at Tsinghua University. I am going to present our work published in PNAS recently. The title is NeuronMotif, Deciphering cis-regulatory code by layer-wise demixing of deep neural networks. First, I will introduce the background. We know that cis-regulatory elements regulate the gene expression. In cis-regulatory DNA sequences, there are many transcription factor binding sites. The corresponding transcription factors can bind to those regions to regulate the gene expression. Different cis-regulatory elements may result in different gene expression level, which determine the function of cell. So deciphering the cis-regulatory sequence grammar is important to understand the gene regulation. We consider cis-regulatory sequence as a natural language, but different from English. It has no vocabulary and no separation between the words, so the grammar is difficult to summarize. In recent years, deep convolutional neural network can accurately predict sequence function, which implies that CNN has already learned motif and motif syntax from the genome sequences. However, CNN is a black box that is hard to interpret, so our target is to interpret the grammar learned by the CNN. From the experience of computer vision, the shallow layer neurons learn simple patterns, and the deep neuron learns the pattern combination. However, we found that most interpretation methods adapted from computer vision does not work well for DNA sequences, especially for deep layers. We found that multifaceted neuron is the main difficulty for interpretation. Multifaceted neuron can recognize multiple patterns, so the interpretation result of this neuron usually contain mixed patterns that is hard to understand. We also found that most deep neurons are multifaceted neurons. In this widely used CNN, we found that mask pooling gives rise to multifaceted neuron. For a neuron recognizing two shift patterns, the feature maps are different for these two patterns, but they will become similar after mask pooling operation. To solve this problem, we can easily class the feature maps of the sequence of two patterns before mixed by mask pooling. The sequence will be grouped by patterns in this way. So, we propose NeuronMotif to implement this process. First, sampling the sequence that can activate the neuron. Second, clustering the feature map layer-wisely and backwardly. Finally, summarize the glossary and the syntax from each cluster. The sequence in each cluster can be stacked into a long position weight matrix. NeuronMotif cut them into segments based on information content and summarize the glossary and the syntax organized in a tree structure. NeuronMotif can successfully decouple the mixed motif patterns. The example of Basset layer 2 and layer 3 are shown here. There are 3 shifting patterns in layer 2 and 12 shifting patterns in layer 3. Existing methods cannot handle multifaceted neuron, so most results are mixed by different patterns. In the task of extract motif from CNN to measure the performance, we use Q-value of the tom-tom to measure the similarity between the found motif and the known motif in JASPA database. In the result, NeuronMotif found more known motifs, and the motifs are matched better to the known motifs. We calculate the 5 cell type ataxic fingerprinting using the genome sequence that can activate the neuron. See figure A and C. The 2 CTCF motif binding sites match the 2 values in the fingerprinting. We also found that some motif syntaxes are cell type specific, which can be supported by the fingerprinting and the gene expression level of the TF. See Figure D. The motif syntax can also be supported by the sequence conservation across species. Finally, I will give the summary. First, multifaceted neuron is the main difficult to interpret neural network black box. Second, mask pooling gives rise to the multifaceted neuron. Third, NeuronMotif interprets CNN through backward and layer-wise clustering feature map of the sequence with different patterns. Fourth, NeuronMotif can be applied to the motif or the motif syntax enrichment analysis and help to understand and improve CNN structure.